Every mitotic and meiotic division is preceded by DNA replication. And in today's video, we're going to talk about the structure of DNA and its replication process, and why the continuity of cell lineages requires that copying of DNA. Hey guys, this is Mikey from Able Prep Academy, and today's video is on that second part of chapter 16 titled The Molecular Basis of Inheritance. This part of the chapter deals more with the structure of DNA and its ability to self-replicate, and it's obviously a very important thing to know for your May exam. Before we get started today, it would be amazing if you guys could like this video and subscribe to our channel. It helps us out a ton in getting our message out there. So let's get started with today's video. In our previous video, we talked about the historical experiments that led up to the discovery of DNA by Watson and Crick. And in 1953, their paper in the journal called Nature laid out the model of DNA that we still understand to be accurate today. The model's power is in its ability to link the structure of DNA to its function as a molecule that can encode data and self-replicate. And so here is that model. DNA, or deoxyribose nucleic acid, is a polymer of nucleotides. So that's a good place to start. A nucleotide is comprised of three components: a nitrogenous base, a deoxyribose sugar, and a phosphate group. While the deoxyribose sugar and the phosphate group remain the same in all nucleotides, the nitrogenous base is where you see the differences. You see, DNA has four possible different nitrogenous bases within its nucleotide structure. You have adenine and guanine, the two purines with the larger structure, and thymine and cytosine, the pyrimidines that are smaller. But like all other true polymers we've seen in AP biology, these nucleotides can link up into a linear molecule. But unlike proteins that we generally model from left to right, DNA is typically shown as being linked from top to bottom. Each nucleotide is connected to the next by a covalent bond that forms through a dehydration synthesis. This bond is called a phosphodiester linkage, and it creates what we call the backbone of DNA. Notice how the deoxyribose sugar and the phosphates bond together, alternating to create this phosphosugar backbone, which is what it's often called. There's also something to mention here that is extremely important. The deoxyribose sugar is said to have carbons that are numbered. The carbon attached to the nitrogenous base always gets the designation of being the first carbon. We then get the second carbon, the third, the fourth, and the fifth carbon in a clockwise manner. The reason that this numbering is so important is that it allows us to establish the directionality of DNA. Up here, where the phosphate is exposed at the edge of this DNA molecule, well, that's the 5' end, because the phosphate is attached to that 5' carbon. Down here, though, where the hydroxide group is hanging on the edge, that's the 3' end for the same reason. So now we have a single strand of DNA, but we know that DNA is actually a double-stranded molecule. And the typical phrasing that we use in biology is that DNA is anti-parallel and double helical. So let's see how that plays into our model. Well, firstly, we have a single strand of DNA that runs 5' to 3' on the left side. On its opposite end, we have another DNA strand, but it runs from a 5' to 3' from bottom up. This is what is meant by anti-parallel, and it also twists upon itself, which is also seen in pretty much all of the DNA models you run into on the internet or in your textbook. But more important is what's happening between the two strands. As Chargaff pointed out, adenine and thymine and guanine and cytosine always occur in equal proportions. And what Watson and Crick predicted using this information was that the bases on the inside of the DNA double helix were paired up in this very specific way, with adenine paired against thymine and guanine paired against cytosine. And this would be true regardless of which base showed up on whichever side of the DNA strand. This complementary base pairing rule is something you absolutely have to know for this course, and to add just a bit more information, keep in mind that these bases are held together by hydrogen bonding, which is typically thought of as a weak intermolecular force. However, with 2 hydrogen bonds between A and T and 3 between G and C, the length of a DNA molecule has so many of these hydrogen bonds that the two strands are held together pretty tightly. Now that we have the structure of DNA out of the way, let's think about why the structure lends to its ability to hold genetic information. In informatics, we can encode information using three simple parts. First, you need the code, then you need the sequence, and finally, you need the direction of encoding. For example, if we were to look at English, we have the 26 letters of the alphabet that acts as the code, not counting for punctuations and capitals and lower cases, of course. But I can sequence these letters, say D, O, and G, and if we all agree that we would read it from left to right, well, I've just encoded and you've decoded a bit of information. The same thing is true of DNA. We have the codes, the nitrogenous bases, and in a long strand of DNA, we can definitely have sequences of those bases that, if read from 5' to 3', would hold information. Now, what this information exactly means will be explored more deeply in the next chapter that deals with gene expression. But for now, we just have to know that DNA is data capable in a digital way. And that's a great thing, because we commonly hear this phrase that DNA is the blueprint of life. With trillions upon trillions of bases in our human genome, we have enough information to basically create a human being. Now, this is of course true of every other living thing as well, from bacteria all the way to bats. But the question then becomes, to what end? Now, I know that it's in poor taste to ask why questions in biology, as if living things have to have a purpose. But if we were to make any comments on why DNA encodes information the way it does, I think most biologists would agree that it's for this one particular reason: reproduction. Whether we're talking about genes that help us grow our hair or allow us to solve complex math problems, these genes survived across time because it helped us reproduce. Otherwise, they would have been lost to time, getting weeded out through the process of natural selection. So now we get to the next and the last part of this chapter. How does that DNA molecule actually replicate? Now, DNA replication is an incredibly complex process, but for AP biology, you don't need to know every single detail. However, you do need to know certain key facts that will help you in deciphering questions on this exam, especially as they pertain to this unit. As mentioned earlier in this video, DNA's complementary bases are critical in its ability to replicate. Let's explore why. Watson and Crick proposed that DNA's complementary bases could be leveraged in its ability to replicate. They call this the semi-conservative replication mechanism, and it looks something like this. Let's say that we take this length of DNA. I'm going to give you 5 seconds to memorize this entire sequence. Are you ready? Okay. Do you remember what the sequence was? Of course not. Without any information on the screen right now, it'll be almost impossible, without rewinding the video of course, to recall exactly what those letters were. But how about now? I'll show you one strand and you tell me what's on the other strand. And now it's easy, and I could have done this with the other strand to ask you to tell me what the top strand would have been. What this simple example demonstrates is the amazing ability of DNA's single strand to hold all of the information required to create its opposite strand. And if you think about it, then it starts to make a lot of sense why DNA is such an incredible replicator. If we split the DNA molecule into two, each of the two strands would contain sufficient data to entirely create the opposite strands, which at the end of the process would give you 2 copies of that exact original DNA strand. And that is why we call it the semi-conservative replication mechanism. DNA replicates itself by keeping one of the 2 parent strands as a template to create a new strand, resulting in 2 copies of DNA, each of which that contains 1 original parent strand and 1 newly synthesized strand. Now, this was just a theory from Watson and Crick's perspective in 1953, but it was actually demonstrated to be accurate by Meselson and Stahl, who experimentally showed this theory's validity. Now, we're not going to go into that particular experiment in this video, but perhaps there may be a separate video on that experiment in the future. Or, you can find one on YouTube. But wait, don't go yet, because now we get to the actual DNA replication mechanism itself. So let's take a look at that process. Whether we're looking at a prokaryotic or eukaryotic cells, the DNA replication mechanism is pretty similar. Looking at the bigger picture in this diagram, we see that the circular bacterial chromosome is about to divide. Fortunately for AP Bio, we don't really need to know what precedes DNA replication, but we just sort of start at the replication bubble that forms at the ORI, otherwise known as the origin of replication. That's an actual region of DNA that has the codes that begins the replication process. Once we have this bubble though, I like to create this middle line that separates the left side of the replication bubble from the right. This is because DNA replication proceeds in both directions, eventually meeting at the end to complete the replication process. But for now, let's zoom in on the left side of the replication bubble. Here we see that DNA replication process has just begun. The point at which DNA continues to get unwound, well, that's what we call the replication fork, which makes sense. But wait, before we get too carried away, there's something we need to mention, which is super important. It turns out that through the process of evolution, DNA has evolved to only elongate from a 5' to 3' direction. This means that the nucleotides can be added only to that 3' end of a growing molecule. If you want to know why that's the case, there's actually a video link in the description below that covers this idea, but for now, let's just say that this is a rule that we have to follow. So let's take a look at what's happening at the top. Here at the top, we have the parent or the template strand of the DNA from a 3' to 5' direction from the midline to the replication fork. And this is good because the new DNA can read this template from a 3' to a 5' while attaching new nucleotide in that 5' to 3' direction on the newly synthesized strand. However, the enzyme that actually does this, known as the DNA polymerase, can't do this from scratch. The DNA polymerase, while really good at adding deoxyribonucleoside triphosphates, can't add anything unless there's a pre-existing 3' end, so we're gonna have to start with something slightly different. You see, primase is an enzyme that begins the actual process of DNA synthesis. It lays down a short RNA primer against the template strand of DNA, again synthesizing that RNA strand from a 5' to 3' direction. Now that we have that 3' end, DNA polymerase can do its thing and place down the correct complementary bases against the bases on the template strand, replicating that DNA the way it's supposed to be done. And in this image, you can see that polymerase can do this quickly, and it's just at the edge of that replication fork. We also see here an enzyme called helicase, which is responsible for unwinding more of that DNA right at the edge of that replication fork. Oh, and the primer that we just put down, well, there's another DNA polymerase that can actually remove that RNA and replace it with DNA, which then would create an entire complementary strand of DNA as the helicase and the polymerase continues to unwind and put down new strands of DNA against the template strand. But there's one major problem. Earlier, we established that DNA has an anti-parallel structure. This means that at the bottom of this picture, where the DNA runs from 5' to 3' from the right to the left, we have an issue. See, while the replication fork moves to the left, its synthesis of new DNA must be happening from left to right, which is the opposite direction to the movement of the entire system. And in order to resolve this problem, cells have come up with a working fix that, while a bit annoying, gets the job done. So here is how that's going to work. Now, everything we've seen so far is the same, except that it happens many times discontinuously on the bottom strand. We have the primase adding the primer, then DNA polymerase doing its thing, and of course DNA eventually replaces RNA. But now the primase goes up towards the more unwound DNA template strand and repeats this process over and over again. Each of these fragments, called the Okazaki fragments, are then later connected by an enzyme called ligase that would create this continuous strand of DNA. So because we have this major difference between the top and the bottom strand, we typically call the top strand the leading strand, while the bottom one is called the lagging strand. Now, what's fascinating though is that if you cross over to the other side of that middle line, then what the leading strand is and what the lagging strand is completely flips. This actually makes a lot of sense, as the direction of the replication fork switches the moment you cross that middle line. So this whole process is incredible, and what's more incredible is that it's super accurate. See, DNA polymerase and the accessory enzymes that are involved in the process continue to check to ensure that errors are not added to the code, which could potentially cause cancer or other harmful mutations, especially if it happens in the germline cells that create sperms or eggs. So even though in today's age we have great understanding of DNA structure and its replication, what's incredible is that it was all sort of predicted in that 1953 paper. It was a really monumental moment in modern biology. And yes, there's still a bit more content left in chapter 16, such as mutations and what actually happens at the ends of the chromosomes during replication, called the telomere regions. But I think those topics are interesting enough for another video, so stay tuned for that one. For now, this has been Mikey with Avo Prep Academy. Remember to like and subscribe, and we'll see you in the next video. Take care.